in the semi-conservative model, here we see you have the two parent strands, and when they undergo DNA replication, each one swiftly gets a new daughter strand represented by a green color. In the next phase of DNA replication, you'll end up with two strands clearly made out of DNA strands that were daughters of the first round, as well as two mixed ones.
We're going to be doing a model of how DNA replication is done in a prokaryotic cell. We're going to be using the semi-conservative method to describe how the parental strands go from parental strands, double-stranded helix, to a separate daughter strand double helix down here. The helix is going to be made up of 4 nucleotides: adenine, thymine, guanine, and cytosine. For also our model, we're going to be using polymerase 1, DNA polymerase 3. We're going to be using gyrase, helicase, DNA ligase, the single-stranding binding proteins, and the new daughter strands that are being made, along with the RNA primers. Alright, so first we start off with the parental strand, and then we come in with the DNA helicase, which is used to unwind the parental double helix. Once down here, we use our DNA gyrase to go in and relax the positive supercoils with negative supercoils. This goes in the left direction. Once that is completed, we go in with our single strand binding proteins to attach to the inside of the helix that is separated to make sure that it does not come back together again. Alright, and then we have to go in with our RNA primers from the 5' to 3' end. So for the leading strand, this will be very continuous. But for the lagging strand, this will be discontinuous, considering that it goes in the opposite motion of the fork, making it harder for DNA polymerase to come in for the lagging strand and synthesize it, wiping out the single-stranding binding proteins, and it makes the primer move over so that it can again do that. For this side, the DNA polymerase is continuous. So it will go straight down the line and the new strand will be synthesized, as you can see by the red. Alright, and then we have DNA polymerase 1 for the continuous strand and removes the primer and then fills in the appropriate nucleotides. However, as we can see in this model right here, for the lagging strand, we have the primer and we have a new term called the Okazaki fragments, making it a gap between the strands in which DNA ligase will come in and bind those strands together, making it one new continuous strand over here in the red. And then once all of that is completed, we end up with 2 new daughter strands from our first parental strand up here.